Would somebody indicate to me or somebody let me know if you can see the slides and hear me talk?
Yes, I can hear you.
Okay, thank you very much. I do appreciate it. Uh, Number of things to go over. We'll start with Pearson here. The new Pearson assignments are up for the next test. I realize we're just now test 2 is opening up right now, but we're starting into the next unit. 9, 10, 11, and 12. 9 and 10 are muscles, and 11 starts the nervous system. 11 is the general overview of the nervous system, and 12 is the central nervous system. So, as I mentioned to you previously, I don't assign many dynamic study modules. I assign chapter 2 and 3, the dynamic study modules, because those were chapters that you should have had in high school or college, and a lot of you probably didn't, or it was a good way to review. Well, of the 5 dynamic study modules that I assign 2 more. Number one's on chapter 9, which has 4 parts. Okay, has 4 parts, and on which deals primarily with muscle physiology. And then the one on chapter 11. Okay, these are required to do. Somebody asked me last time, were they required? And the answer is yes, they are required to do. Okay, so you have 2 more study modules this time. You didn't have any on the skeletal system because I didn't feel like there was anything that was in that much detail. In muscle, it's primary muscular contraction, and in 11, it's primarily nervous generation of action potential. Number of those have a bunch of steps. Okay, and then you have the chapters test on 9, 10, 11, and 12. So you have 4 and 4 dynamic study modules, that's 8, and then a chapter test on 9, 10, and 11, and 12. So I just wanted to go over that so everybody would be clear about that. Next is the assignments. If you read my notice, my announcement, I was not able, despite everything that I wasted a lot of time trying to get, excuse me, I gotta get something to drink. Trying to get the slides converted to a Word document. And the problem was they just weren't clear enough that I wanted you to try to guess what the structures were on a Word document. So we're going to do the lab practical. Okay, lab practical, which opened up and is going to be available till October 29th. We're going to do that just like we did the tissues. You have the slides, you have an answer sheet, okay, and you have your list. There is no time limit for taking the exam. Okay, so there's not going to be a 3-hour time limit. There is no time limit, okay, for taking the exam. There is a date that is due, which was 2 weeks from Thursday when it opened. Okay, so 2 weeks from last Thursday, it will be due. We're going to do the skeletal system lab practical, and then consequently the muscles and the nervous system special sensation. We're going to do those all the same way, just like we did the tissue. So it's not going to be a timed exam. I will work on trying to do a time exam over the Christmas holidays when I have some time to do that. Uh, so that is what's going on there. So the lab practical and skeletal system is you have your answer sheet, you have the slides, and so you can do it. You can do part of it, stop, do some more of it, and you have your list that you should have made out. So that's the lab practical. The exam 2 is going to open up today. Okay, exam 2, which is on chapters 5, 6, 7, and 8. The skin and then the bones. Okay, similar to the other exam, matching multiple choice, true, false, and short answer. It will open up today and at noon, and it's due in a week. So I just wanted to go over that. Exam 2 is going to open up today at noon, like I told you it would. I said it's going to open up Monday, and today's Monday, so it'll open up Monday, and it's due in a week. And otherwise, I don't think I didn't put up your week 8 yet. I'll get that up But I don't think, except for the exam, which you'll be due in a week, I don't think there's anything else that's due except for the Pearsons associated with the exam. Is in regards to modules, okay, in module 8 and 9, it's another combined one, or excuse me, 9 and 10, another combined one. You have your lecture notes, the PowerPoints that we're going to go over, the recorded lectures from last semester. These are the Pearson PowerPoints for 9 and 10 if you want to look at them, okay? For 9 and 10 if you want to. In regards to the lab is going to be, number one, you have one for the muscle, you didn't have for the bone, but you have one interactive physiology, okay? And that's just a matter of watching a few little videos and putting the answers into that answer sheet. You have a list of muscles here. This is the list you would get if you're doing a face-to-face. So you have a list of muscles here. So this is your muscle list. So what I want you to do is start going over these muscles. Okay, it'll be the list that you're going to use when you take the exam. And then you have the answer sheet here. I have not opened up the slides yet because I want you to be working on the skeletal lab practical. Well, I'll probably open these up next Monday after, because that's because we're going to take 2 weeks to do this. And so we'll probably end up doing the lecture today, the lecture on Wednesday, going over the muscles, just like we did last Monday for the skeletal system, and then having that Wednesday off. So I'll open up these slides next Monday for the muscle lab practical. And so all this is open except for the slides. And obviously all of this is open. I'm getting better about opening things up. So you have your recorded lectures, you have your PowerPoints, you have the lecture notes, and you have the Pearson slides if you want to do those. So I wanted to go over all that to make sure that we were all on the same page and knew what was going on. So we're going to start on the anatomy of the muscle. I don't know how long my voice is going to hold out. But we're going to start on that. And then we'll get as far as we can. And then whatever we don't get to, it's not a whole lot of slides. We will spend some time talking about muscular contraction, spend some time today if we get to it. No, we won't get to it today, but we'll get to it on Wednesday and probably review it on Monday, because there's two hard things in this unit. One is muscular contraction, the interaction of actin and myosin, and the physiology associated with that. And then, like I say, with the nervous system in chapter 11, the action potential. So we'll go over it in lectures and we'll revisit it next Monday again to make sure that you have a firm grasp of that. So the muscular system, functions of the muscle tissue, obviously is movement, maintaining posture, stability, and heat production. Remember, if we get cold, we shiver. If we get cold, we shiver, and that generates heat. Comparison of muscle types, universal characteristics, they're all excitable, okay? Muscle fiber is just, it does one thing, it shortens. That's all a muscle fiber does, it shortens. If it gets a stimulus which causes it to contract, it will shorten. That's all it does. It just shortens. It doesn't generate anything, just happens to be attached to other bones, and because it's attached to other bones, when it shortens, it's going to make movements. Okay, it has the ability to shorten. That's what happens when you get a action potential down to there. It's able to be stretched. Okay, because we can stretch our muscle fibers when we contract the antagonist. Okay, we have an agonist which does the major muscle movement, and we'll get to those words. We have an agonist which does the major movement. So if we wanted to bring our forearm, okay, our antebrachium up to our brachium, up to our arm, we would contract the biceps. And so that muscle the biceps has shortened, okay? If we want to extend our antebrachium, our forearm, then we would contract the triceps, okay? When we contract the triceps contracts, but the biceps would extend, okay? Biceps would extend. And then elasticity, ability to return to the resting length. Skeletal muscle, we'll review the muscle types again. We did it once before. We're going to review them again. When you had tissues, you know that the skeletal muscle were long, unbranched cylindrical fibers. They were multinucleated. They had a bunch of myofibrils, and those myofibrils were those beautiful striations that we talked about, very regular pattern. They have an extensive tubular system. We don't call it endoplasmic reticulum. In a muscle, we call it sarcoplasmic reticulum. It's the same thing. But in the muscle, it does a little bit different function, and we call it sarcoplasmic reticulum versus endoplasmic reticulum. We have T tubules, and we'll talk about those. It is striated. It is voluntary. It's the one that you and I control. And most are attached to bones because that's what we want to do with them, is do movement. And this is our skeletal muscle that we remember from our tissue slides, okay? Cardiac muscle. Cells are branching and cylindrical. They are uninucleated as opposed to multinucleated, less well-developed sarcoplasmic reticulum, fewer myofibrils, more mitochondria. It's mitochondria, but they call it mitochondria, and that's actually the only time I've ever seen that term. In all my studies, undergraduate and medical school, and everything I've ever studied, I never saw that term mitochondria until I looked at these slides 5 years ago. But mitochondria or mitochondria, more mitochondria, okay, because the heart's got to be, our skeletal muscles don't have to contract all the time, but the heart's beating all the time. Lipid and glycogen deposits, okay. We need glycogen because we've got to have, glycogen is our source of instant energy in animals. Starch is a source of energy in plants. Glycogen is a source of energy in animals. They are striated with intercalated disc. Remember, we talked about that. We want one muscle to connect with another muscle so that they can do contraction. They are self-stimulating, okay, self-stimulating. They generate their own impulse. They're involuntary, and their location is in the heart only. And so these are cardiac muscles, okay? And finally, smooth muscle. Slender spindle cells and uninucleated. So cardiac muscle is uninucleated, smooth muscle is uninucleated, okay? But voluntary muscle is multinucleated, limited number of gap junctions, okay? Skimpy sarcoplasmic reticulum, myofilaments less regularly arranged, so not striated, involuntary, located, mostly found surrounding hollow internal organs like the GI system and blood vessels, okay? And some of the respiratory tracts. And here's smooth muscle here, okay? Anatomy of skeletal muscle, structural organization. We have coverings around the muscle. We have coverings around the muscle, okay? We have three coverings. We have an inside covering, an intermediate covering, and an outside covering. The covering around the entire muscle is called the epimysium, okay? Covering around the entire muscle is called epimysium. A covering around an individual muscle cell, okay, is called an endomysium. This is an endomysium here. These are myofilaments of this individual muscle cell. This is an endomysium here, okay? It's the covering around an individual muscle cell, and the covering around a fascicle, which is a group of muscle cells. A fascicle, which is a group of muscle cells, is called a paramecium. So we have an epimysium, a paramecium, and an endomysium, okay? And we'll see that again when we look at nerves, okay? We have an epimysium, which is the covering around the entire muscle. Notice the muscle is attached to bone by way of a tendon. Ligaments attach bone to bone. Muscles are attached to bone by way of tendons, okay? We have an epimysium, a paramecium, and an endomysium. The endomysium surrounds a number of myofilaments. A muscle cell is composed of myofilaments. Fascicles, a bunch of muscle cells. This is a fascicle here, and it has a paramecium surrounding it. and an epimysium surrounds the whole bundle of fascicles. So it surrounds the whole muscle. And you can see these. These are white fibrous-like materials, okay? They're not real thick, okay? But you can see that. These are ways of categorizing muscles, just like you can categorize bones, large bone, long bones, short bones, cube-shaped bones, and irregular bones, okay? Well, we can have parallel fibers where the fibers run parallel. We can have fusiform, which is thick at the middle, okay, and tapers on, fusiform, which is thick at the middle, okay, which example would be the biceps, and tapers on both ends, okay? That's a fusiform shaped muscle, biceps brachia. We can have a convergent, which is thick attachment at one end, okay, tapers to the other, and that's the pectoralis major. We can have pinnate, okay, which are feather-like. These are pinnate. This is multi-pinnate. This is bipinnate, and this is unipinnate. These are feather-like looking muscles, okay? We can have circular muscles that run around in a circle. We have some in our lips called the orbicularis oris, okay? And we have one around our eye called the orbicularis oculi. Muscle attachments. Muscles can be attached directly to the bone, okay? So here's the temporalis muscle. This muscle is attached directly to the periosteum of the bone. We can have indirect tendons that attach muscles to bone, actually penetrating the bone. This is example here, the masseter muscle. Okay, the masseter muscle has a tendon which is attaching to the bone, temporalis right here, attached directly to the bone. And then we can have an aponeurosis, which is this is a thick, this aponeurosis is thick, okay? If you do incision across the top of the skull, you go through the skin, you go through the aponeurosis, and then you go to the bone, okay? We have muscle attachments. So the frontalis muscle and the occipital muscle actually attach to the aponeurosis. They attach to the aponeurosis here. So we can have a direct attachment to a bone. We can have attachment to a tendon to a bone, to a tendon to a bone, and then we can have an attachment to an aponeurosis. And so those are three types of attachments. Microscopic anatomy. Sarcolemma, remember we don't, in muscle, we name things different for whatever reason. So rather than having a plasma membrane, we have a sarcolemma. Rather than have cytoplasm, we have sarcoplasm, okay? And the muscle just happens to attain, what's the difference between not regular cells, but non-muscle cells and muscle cells? Muscle cells store glycogen, especially in the voluntary muscles, and super especially in the heart, okay? Super especially in the heart. So we're looking at a muscle cell, okay? We're looking at muscle cell, and this is one of the myofibrils, okay? This is one of the myofibrils. This is the mitochondria here. So you can see they have a lot of mitochondria there. If we're looking at muscle cells with all these definite striations, this has to be voluntary muscle, okay? We have mitochondria, which is ATP source. We have myoglobin, which is a hemoglobin-like molecule that carries oxygen in the muscles. We have myoglobin, which is a hemoglobin-like molecule that carries oxygen in the muscles. And so we're going to look at the microscopic anatomy of an individual myofibril, okay? A sarcomere is the basic unit of a muscle cell, and it extends from the Z disc to this Z disc, okay? This is a repeating structure. These are the red filaments, are the thicker filaments called myosin, and we'll look at these. And the blue, I'm just giving them a name. And the other filaments, the blue filaments, are called actin, okay? And these are responsible for contraction. And the reason we see these striations is the overlap that we have, So the basic structure is a Z disc to a Z disc, okay? Then we have an I band, okay? We have an I band, which is this part right here, okay? This part right here. We have an A band, which is this part right here, and then an I. So it repeats I, A. And then we have a Z disc, an H zone, and an M line. A Z disc, an H zone, and an M line. So we have 6 parts here. 6 parts here. And on a face-to-face test, you'd been given a picture of this and you'd have to explain what they are. Okay. Didn't have to draw it, you just have to explain what they are. And so these are the parts. These are the parts of a sarcomere. Sarcomere is the fundamental unit of a muscle cell. It extends from one Z disc to the other Z disc. We have repeating units, I band, A band, I band. Okay. You can see that the I band contains only the thinner filaments. The A band, portions of it, the A band contains the thick filaments totally all the way across, and then portions of it contain thick and thin. The H zone only contains the thick filaments, and the M line is where the thick filaments attach. And the Z disc, this actual disc right here, is where the actin attaches, the thin filaments. And we're going to go over that in detail, okay? This is what it looks like in much more detail, okay? This is a Z disc to a Z disc. This is your thin filament, thin filament, thin filament. The blue one is your thin filament. That is called actin. This red one is your thick filament. That's called myosin. Notice myosin has heads. Okay, it's a long, thick filament which has heads. So if you can imagine during muscle contraction, there are binding sites on this actin that are going to be exposed, and we're going to go into detail as how they get exposed. Once they get exposed, these heads of the myosin are going to attach to the actin, and they're going to move in this direction. They're going to do a stroke-like movement, okay? And these heads will move in this direction towards the M line, and these heads will move in this direction towards the M line. And when it does, it causes the Z disc to come closer together. And so that's how, so this is an individual sarcomere. Imagine millions of sarcomeres contracting. And when that happens, then you're going to have muscle shortening. Okay. So when that happens, you're going to have muscle shortening. Notice we have the thin actin filament. We have elastin, which is titin filaments, which attach the thick filament to the Z disc, okay? The thin filament's already going to be attached to the Z disc, okay? But the thick filament is going to end at the A band, and so we have fibers, elastin fibers, which come out and attach it to the Z disc, okay? So we have a Z disc, we have a I band. The I band, we don't see it all, but it would go from here to this end. That's the I band. The A band goes from there to there. Okay. The H zone is where the actin ends here. So this is the H zone here. This is the H zone here. And this is the M line. And the M line is where the myosin attaches in the middle. You know, for it to have enough grasp, enough stabilization, it's attached here, the Z disc, and attached here at the M line. Okay. So it'll just, all this is just anatomy. It's just a nice, pretty picture. And they have electron micrographs that this is what it looks like. And so you're just going to have to go over this a couple of times till you get an idea of what this is. Okay. Because you could have a question, what is composed in the I band? Well, in the I band, there is only actin. Okay. In the I band, there's only actin. There is no myosin. Myosin is only in the A band. Well, what's in the H zone? In the H zone is from here to here. That is where there is no actin. The reason there's no actin is we're going to pull the actin in that direction, So this is this gap that actin is going to shorten in. Okay, so that's why there's no actin there. If there was actin there, then it would bubble up. It would overlap, and we would. And that's not the way it happens, okay? Microscopic anatomy. The myofibrils contain the myofilaments, actin and myosin. Actin is a thin filament. This consists of both tropomyosin and troponin. Okay, it consists of both tropomyosin and troponin. The myosin is a thick filament. Tentin is elastic filaments that secure the myosin to the Z disc. And so we're looking here at actin. Okay, we're looking here at actin. Actin is we'll go over the anatomy more in a little bit. The A band is the area where there is myosin. So where we have the thick filaments, which is the red filaments, that's the A band. This is the A band from there to there. The I band is where there is only actin. So here is the I band from here to here. Notice we only have the blue filament, which is a thin filament. That is our actin. Okay. The H zone is the area where there is no actin. So this is the H zone. Okay. This is the H zone from there to there. H zone from there to there. From there to there. There is no actin. There's only myosin here. The Z disc anchors the connexin. Okay. The Z disc composes the connexin, anchors thin filaments. This is connexin, anchors thin filaments, and connects the myofibril across the width of the muscle cell. So it connects both the actin and the myosin. The myosin, because it ends, has to have elastic filaments, elastin to connect it. But all these filaments are connected here at the Z disc, and the myosin is connected at the M line. Okay, so this is an electron micrograph. Okay, electron micrograph showing you the Z disc from there to there. The I band, the Z disc is here. This is the I band. This is the A band. Okay, this is the A band. Now, wouldn't this have been good when they were naming it if they would have named it the I band, the A band, because A band only has actin, and named the A band the M band because that has myosin. Okay, so we have the Z disc, we have the I band, we have the A band, we have the H zone from here to here. This is the H zone from there to there. There is only actin, there's only myosin, there is no actin, and this is our M line right here. This is the M line right here. And so between two discs, we just keep going into over and over again. This is the same picture. This is a sarcomere. The functional unit of a myofilament, myofibril, is from one sarcomere to the other sarcomere. Okay. The sarcoplasmic reticulum is smooth endoplasmic reticulum. It is not rough. It's smooth endoplasmic reticulum. The major function is to store calcium. The major function is to store calcium. This smooth endoplasmic reticulum has parallel fibers that are going to surround each one of these filaments. So the parallel fibers are going around each one of going down along each one of these filaments here, myofilaments. We have a cistern. Okay, cisterns are collecting areas. If you've ever been to people that collect water, okay, collect water. They may have a cistern, or people that have pumps may have cisterns where water is collected. Okay, this is a cistern here, interconnecting tubules that surround, okay, the myofibrils. These are cisterns. Area where sarcoplasmic reticulum runs perpendicular at the AI junction on the muscle. This is at the AI junction. So this would be at the AI junction. T-tubules are tubules that penetrate into the cell's interior. These are T-tubules here, okay? T-tubules that they're going down. The sarcoplasmic reticulum goes outside the cell. The T-tubules are actually going to take the message down into the cell. There is a triad of T-tubules sandwiched between the two cisterns. Okay, so there's a triad of T-tubules between the two cisterns. So we have this triad of T-tubules between these two cisterns here. Okay, we have a troponin tropomyosin complex. Okay, Actin is made up of these little actin subunits. Actin is made up of little actin subunits, okay? Actin is made up of little actin subunits, and they're made in, looks like pearls, okay? They're made in pearls here, okay? And so you put 2 strands of pearls and you twist them in a helix. You twist them in the helix, and then you have tropomyosin. Okay, you have tropomyosin, which I look at as a piece of wire or a piece of duct tape or something like that is going to hold those 2 pearls in line. Okay. The tropomyosin. So we have the actin subunits, which are these small subunits that are put together like 2 strands of pearls. And the tropomyosin goes along the outside of these pearls, goes along the outside of the pearls, and they're going to maintain the integrity of the pearls. The tropomyosin is going to maintain the integrity of the pearls. The troponin, okay, tropomyosin, troponin, the troponin, I look at that like a U-bolt, something that's holding that tropomyosin down, okay? And by holding the tropomyosin down, it is covering up, by way of holding the tropomyosin down, it is covering up the active binding sites on the actin. Remember, we looked at the myosin, we looked at the thick filaments, and it had heads, okay? They are going to bind to the binding sites on the actin, okay? They're going to bind to the binding sites on the actin. But if the muscle's not contracting, the binding sites are covered up. And they're covered up with the tropomyosin, and the troponin is holding the tropomyosin in place. The troponin is holding the tropomyosin in place. The tropomyosin is providing structure to the actin subunits. It's a set of it's 2 strings of pearls that are twisted in a helix. Tropomyosin's like duct tape. I look at it as duct tape or a metal wire, whatever you're looking, to provide structure to it, to provide strength to it. But it also is covering up the active sites, and the troponin makes sure that these active sites covered up. When an appropriate stimulus comes along, this troponin is going to undergo a configurational change and going to release that tropomyosin. It's going to fall down and expose the active binding sites so the myosin can bind to the actin. Okay. We haven't got that far. That'll be on lecture on Wednesday. Okay. Troponin binds the 3 polypeptide complex that binds tropomyosin and actin, requires calcium ions and an inhibitory substance that binds to the actin. The tropomyosin spirals around the actin to stiffen it. For the troponin, just remember that it's the one that holds the tropomyosin in place, and it's the one that's going to interact with the calcium and with the ATP, interact with calcium and the ATP to be able to expose the binding sites. Physiology is skeletal muscle fibers. We have a nerve, okay, whatever nerve it is, going down to this muscle fiber, okay? And the nerve and the muscle don't actually touch. The nerve and the muscle don't actually touch. The nerve's called an axon. It's going to go down to the muscle fiber, and then it's going to end in a motor end plate. It's going to end in a ball-like structure, but there's going to be a gap. There's going to be a gap between the end of the nerve and the muscle fiber. Okay, and then here's our muscle fiber. Most nerves that innervate muscles don't innervate them at one end. They usually innervate them in the middle, because remember, a signal is not going to go down the whole muscle. All the muscle fiber does is contract. So the innervation for a muscle, we're going to talk about this a whole lot more detail. This is just sort of an overview. All the muscle fiber does is shorten. That's all it does. So we don't need to have the nerve at the end of the muscle fiber. We need to have the nerve, or the nerve turns out to be in the middle of the muscle fiber, because all the muscle does is shorten. And so we have a synapse-like relationship between the motor neuron that's going to send a signal to the muscle cell to contract. So here is our ending. Here's our ending of the nerve here. Here is the gap, the synaptic cleft, and here is the muscle, okay? So here is the end of our nerve, okay? There is a gap here. There's a gap here. It's the same thing between two neurons in the nervous system. There's a gap here called a cleft, okay? And it's the synaptic cleft because this is a synapse between the nerve and the muscle, okay? This is the end plate, okay? And in the end plate, they are these vesicles, okay? There's these vesicles that store acetylcholine. Acetylcholine is a neurotransmitter. We're going to study. This is the only one we're studying the muscle. When we get to the nervous system, there's 3 or 4 or 5 more, maybe 4 more, 5 more we're going to study. But in the muscle cell, acetylcholine is the neurotransmitter that is responsible for taking the signal from the muscle, from the nerve to the muscle. And it's contained in these synaptic vesicles. Well, how did it get there? A signal went out from the nucleus to the endoplasmic reticulum. This acetylcholine was made. It was then sent to the Golgi apparatus, which is packaged up and in vesicles waiting to be delivered on the appropriate signal, okay? When an action potential triggers their release, so an action potential is going to trigger their release, the acetylcholine diffuses across to the sarcolemma. What's the sarcolemma? That's the plasma membrane of the muscle cell. Okay, remember, we don't call it a plasma membrane, we call it a sarcolemma, which contains the receptors for the acetylcholine. Okay, it wouldn't make any sense if we released acetylcholine and the muscle didn't have any receptors. So when the action potential is triggered, we get release of the acetylcholine. The acetylcholine is going to come across the synaptic synaptic cleft, okay, and interact with the receptors for acetylcholine, okay. The motor end plate membrane on the muscle generally center that contains the receptors. So what this is saying is just what I said a while ago. The motor end plate, rather than being on one end of the muscle, is generally in the center because all the muscle is going to do is contract, okay? And it contains the receptors for acetylcholine that will depolarize. And so the muscle, the sarcolemma, is going to retain the receptors for the acetylcholine. That's going to depolarize, okay? We'll get a depolarization in nervous system. But in other words, the signal is going to continue from the nerve to the muscle, okay, and send the signal deep into the muscle cell, okay, because we want all the fibers to contract. We don't just want the fibers on the most superficial outside to contract. We want them all to contract. Mechanism of contraction generating an action potential. Action potential travels across the sarcolemma and down into the T tubules, okay? We have this triad here, okay? We have the T tubules. The action potential is going to travel across the sarcolemma and down the T tubules. It's going to cause the release of the calcium ion, which is stored right here, okay? It's going to cause the release of the calcium ion, which is stored right here in the T sarcoplasmic reticulum. And in the T tubules is where the calcium ion is stored. Okay, the calcium ion is released. Calcium binds to troponin. Okay, calcium's going to bind to troponin, and we're going to get contraction. Calcium's going to bind to troponin. Remember, we have tropomyosin, which is that long outside strand, which provides the stiffness. Has to, the actin subunits are in, it looks like pearls to me. They're in 2 long lines of pearls. They're twisted into a helix. The tropomyosin is along the outside there to provide stiffness to it. The troponin is the one that holds the tropomyosin in place, covering the binding sites, okay? The calcium is going to bind to the troponin, and that is going to cause a configurational change, exposing the active sites, and this myosin head is going to attach to an active site. Myosin head is going to attach to an active site, and contraction is going to be in it. Okay. A muscle at rest. I bands and H bands are at their longest length. Okay. I bands and H bands are at their longest length. Sarcomere is at its long length. Calcium is in the sarcoplasmic reticulum. Troponin, tropomyosin complex covers the active sites. ATP, some stored ATP, but only a few seconds worth once the contraction starts. Okay. So this is just a schematic. This is a schematic of what's going to happen. These receptors, okay. And we have these vesicles here, which are holding acetylcholine. When we get a signal here, when you get this action potential, is what the signal. The action potential is going to go down the nerve, and we're going to study that in detail when we get to the nervous system. But the action potential is just a signal that goes down the nerve. Signal's going to go down the nerve. Once it gets to the end here, these vesicles are going to be released. The vesicles contain the acetylcholine. The acetylcholine is going to fuse across here and interact with the acetylcholine receptors here. Acetylcholine receptors are going to be interacted, going to start a depolarization in the muscle cell. Depolarization in the muscle cell is going to go down deep into the muscle cell. It goes down deep into the muscle cell. It's going to cause calcium to be released. Okay. Calcium is stored in the sarcoplasmic reticulum. The calcium is going to interact with troponin. Okay. Calcium is going to interact with troponin. When it interacts with troponin, and like I say, I look at troponin as a U-boat. It raises up the U-boat and the U-boat turns, but technically the troponin undergoes a configurational change, and it lets the tropomyosin slip down and expose the active binding sites. And the myosin head, after it's been primed with ATP, is going to attach to one of the active binding sites and going to generate going to generate a pull. Okay, it's going to generate a pull, which is going to cause the actin to slide over the myosin. The myosin's not going to change length. The actin's not going to change length. It's just going to slide over the top of the myosin. And so that's the sliding filament theory. And this is about halfway through the slide, so we're going to leave it here. We're going to go back over the stuff that I talked about early on. Number one, the lab practical was opened on last Thursday. I was not able to get it into a configuration that I was happy with, that I felt would be fair for testing you in a Word document, because the slides were just not as clear as they are on the PowerPoint. So we're just going to do the skeletal system just like we did the tissues. It is not a time test. You can start it and stop it. There is a time limit to take the test 2 weeks after open last Thursday. So it has to be turned in by 11:55 on that day, but there is not a time limit to the test, okay? But there is a date due to the test. Exam 2 opens today at noon in about 3 hours and 15 minutes. It is similar in format to the last exam, multiple choice, matching, true, false, and short answer. It covers chapters 5, 6, 7, and 8, okay? 5, 6, 7, and 8. It is due 1 week from today at 11:55. So exam 2 will open today at noon. You can find it under assignments. You just click on it. But remember, once you click on it and start it, you only got 2 hours to finish it. Okay. So you want to make sure you're ready to do it. You're not in the middle of a thunderstorm. You're not going to lose your internet connection, anything like that. And it's open book, open notes. But I will remind you that some of you came very close to plagiarism last time. And if I find any similar configuration to anything outside the book and the notes, okay, remember it's the Dr. Oler's notes, the slides, whether it be our slides or the Pearson slides, the book, okay, anything I might say in lecture. If you go to Khan Academy, if you go to Wikipedia, if you go to anything else and you copy and paste or you paraphrase is the same thing, okay? How do you get around that? because when people start talking about stuff that I didn't talk about, okay, that I didn't talk about in the notes, or that Dr. Oler didn't talk about in the notes, I didn't talk about in the lecture, and they go into depth, they go into the depth that you get in medical school, then something's a little bit fishy on that answer, okay? Also, remember, if you have 10-point questions and you answer 2 sentences, you're probably going to get a point, all right? If it's a 10-point question, that means you need to write a little bit, all right? And so if it's a 10-point question, you need to write a little bit. Lastly, the new Pearson assignments open up. You know, this for exam 3, it's going to be 9, 10, 11, 12. They are 4 chapter tests, 9, 10, 11, and 12. They are 2 dynamic study modules for chapters 9, okay, for chapters 9, a lot of which is this sliding filament theory and action potential and things that we're going to generate, that things that are generated, you know, that you need a little extra learning mechanisms for. And chapter 11, which will have the same thing, but we'll talk about in a nerve, which is much more complicated. Well, it's not more complicated. We just talk about it in more detail with the sodium and potassium channels and depolarization, hyperpolarization, things like that. So you have 2 dynamic study modules, one for chapter 9, one for chapter 11. It has 4 parts each. So the total number of assignments on Pearson, are 4 plus 4 is 8, plus 4 chapter tests is 12, okay? The lab module for this time for muscle contains 1 interactive physiology, which is just where you watch the little videos and then fill in a chart. Yes, it's redundant. Yes, you have to do it multiple times, but this helps you to learn. Remember, you should be taking a face-to-face test, but we don't take face-to-face tests. I still don't do proctoring. And so you have a little bit of an advantage, but you need to learn this material and so repetition is the key on material to learn. And so you have that, and then you're going to have a lab practical. What you have is your you have the muscle list in alphabetical order, which would be the same muscle list I would have given you if you were doing a face-to-face exam after we'd have spent 2 labs on the muscle, sometimes 1 lab, 1 or 2 labs on the muscle. So you have that word list, so you can start going through that word list and looking at your muscles if you don't have anything else to do, including the skeletal practical and the test, but you know, it's there. We will finish up the lecture on Wednesday. On Monday, we will go back over how a muscle action potential and muscle contraction occurs, because that's something that we're going to be repeated. And the structure of and the striation structure, we'll go back over that. And then we'll do like we did on the skeletal system. We'll talk about the individual muscles, and I will run through the muscle lab practical, and I'll open the muscle lab practical next Monday, and you'll have 2 weeks to do that. And so that's pretty much it. You've on your module thing, you've got Dr. Oler's notes, you got the PowerPoints, you got the recorded slides from last semester, and you've got the PowerPoints from chapter 9 and chapter 10. So you pretty well much have all the information you need to start this unit. And I don't have anything else to say. Does anybody have any questions? But if you don't have any questions, we're done. On Wednesday, we'll finish up the lecture. Don't forget that Don't forget that the lab practical is open for the skeletal system, and the exam 2 opens today. You've got a week for exam 2. You got 2 weeks from last Thursday for the lab practical. All right. Thank you very much. I appreciate it.